My name's Jolene, so I'm with SNEV on the events team. Um, and we have Jonathan and Barbara with us today as well. So we'll each take a turn introducing a speaker, and then we'll guide you guys through the Q&A after. So our first speaker is Inês. She's currently at the University of British Columbia working as a postdoc in Dr. Michael Hayden's lab. And I'm not going to share too many spoilers because I'll let her share her journey, but she started in Portugal. She's been to Germany, she's been to the US and Florida, and now she's in Canada. So she's been all over and has a good breadth of experience to share. So thank you, Inês.
Thank you so much for the nice introduction, Jolene. My name is Inês. I'm originally from Portugal. It's a very difficult name. I didn't know that until I left Portugal, that it's not so easy to pronunciate in English. Uh, Also very hard to write. So I will just talk a little bit about my journey in science and then just give you a little bit about the current work that I'm doing. So I'm originally from Portugal, and I'm from a small city near the coast called Figueira da Foz. This is a photo from my city. So we have a lot of beach and nice weather, a lot of wind too. Um, I don't know how much you know about Portugal. I did my bachelor degree in Coimbra. So it's one of the oldest universities in Portugal. Of course, that we have Cristiano Ronaldo and the hectares that most of you might know. This is a castle in my city, so you can get a beautiful view. And this is Lisbon, and this is Coimbra. Is where you have the tower and the old university. And this is the costume that we also a lot of you might notice from Harry Potter. So the outer base hair. The Harry Potter costumes on the Portuguese universities. So we have the cape, everything is in black. So I did my bachelor there in biochemistry, and at the time I was thinking going to industry. So I then moved to Lisbon to do my master's in biopharmaceutical sciences. But during my master's, I got the taste of science. So then I decided, okay, maybe I will do a PhD and keep my career in academia. And it was when I moved to Germany, to a very small city called Göttingen, where when you graduate, you need to climb this statue with a hat that it's not fixed to your head. And you can let it fall because it's bad luck. So you need to climb it still with your head. And it's the Gänselisel. So it's a lady with a duck, and it's good luck if you climb it. It's from far away, and I never heard about it, but it's the city in Germany that has more than 50 Nobel Prizes. So it's quite amazing for such a small city, and it has everything. It's very good for. It has the Max Planck Institute, the university. So I really loved it there. My biggest challenge was to learn German, so it doesn't influence my work in science, but my personal life, right? Going to the finances, going to the supermarket. So it was a very good experience. It was the first time that I left my country. I learned a lot. And after I finished my PhD, it was in neuroscience, and it was when I started to work with extracellular vesicles. And at this time, I was just looking, how can it be that different proteins related with neurodegenerative diseases are transferred from cell to cell using extracellular vesicles? And it was very based on molecular biology. And when I finished, I actually wanted to move more into biomarkers and therapeutics, be more involved in the clinical side. And it was at this point that I decided to move to the United States. I moved to Orlando, Florida, where a lot of crazy things happen, but it's also a beautiful place. Dangerous because we have the alligators, but it's beautiful because it has the beaches. We have Disney and Universal, all the parks. A bit hot because the temperature is there, but I loved it because I had the experience of starting a clinic in Huntington's disease and interact with patients, know more about their day to day, what they need about what they know about therapeutics. So it was something that sometimes in Europe it's difficult because there are so many regulations that sometimes us as a postdoc or as a student are not really involved in the clinic side of research. So I really love this part in moving to the States. I learned a lot, and it's a big contrast doing research in Europe compared with doing research in the United States, not only in funding and regulations, access to human samples. Um, but my university there was more focused on undergrads, so the number of postdocs was really small. So I felt the university didn't have enough courses or didn't give me enough support to for me to pursue my academic career. That it's my goal is to one day, to have my lab, be a professor. So then it was when I decided to move to Canada in Vancouver, especially Vancouver. So I love Vancouver. It's I didn't know about it until a friend of mine told me, and it's a beautiful place. You have the mountains, you have the sea, you have activities that you can do in the summer and in the winter. The university, it's amazing. The people are very, very sweet. And I love it here. So it remembers me a bit like Europe. But I think that the university really cares about you as a postdoc to progress and to develop and eventually get your own independent career. So this has been my journey. And now I will just talk a little bit about my work. So I work in extracellular vesicles in Huntington's disease. And what I have been doing, I don't know how much you know about Huntington's disease, but this is a neurodegenerative disorder. And when one of the parents has the disease, so it has an expansion on the CG expansion in the Huntington gene, and when one of the parents is affected, there is 50% chance that their kids will have the expansion. There are also some de novo cases. And the main hallmarks is the accumulation of this protein in neurons and a loss of the MSNs and also the striatal tissue. So, and the patients present a little bit like Parkinson's, Alzheimer's, and ALS all together. So it's like a mix of all the different disorders. And we all thought that it was really easy to cure because we know the cause, right? So it should be easy to target. But the problem is there are so many proteins that also have this repeat, and also the quantity of lowering of this protein. So it has been a lot of challenges. So we know what is HD and what is the Huntington protein for 32 years, and we still didn't manage to cure this genetic disorder. And currently, the biomarkers that we use in Huntington's is the measurement of this protein in the CSF by different sensitive techniques. And the problem, so it's very good because it's the only biomarker that we currently have available. And we know that the measurement of this protein in the CSF correlates with the levels in the brain. And we know that in animal models, if we lower Huntington in the brain, it also reduces in the CSF. The problem is not everyone actually wants to go and do lumbar puncture, especially controls. Most of the controls are family members of the HD patients, and you have risks. You can have headaches, you can have infections. So it's very difficult sometimes to get people that are available to give a CSF sample for studies or for clinical trials. And we also know that this protein is present in the plasma, but there is no correlation with disease progression. That is why I started my work during my PhD, and then my first postdoc was to look at Huntington in the vesicles, to see if we only look at vesicles that come from the brain, if then the levels of Huntington in the vesicles from the brain correlate with the Huntington levels in the brain. And I will not go into a lot of details, but a lot of studies have been always focusing on exosomes or small vesicles. And in my work, what I saw is that these vesicles, the large ones that we used to call ectosomes, that burst from the plasma membrane, these ones have most of the quantity of Huntington. So maybe, if we look only to the small ones, we are having problems of detection because some people see it, some other people don't. So maybe if we look at other vesicle types, maybe these are proper biomarkers, or maybe they have the probability to be possible biomarkers for this disorder. And what I'm currently doing is to use human samples. So I do both plasma and CSF. And I try to correlate the levels of Huntington in the vesicles from the plasma and also measure the protein in the CSF and see if it correlates, since we know that the protein in the CSF correlates with disease progression. And here I'm just showing how it looks like. So I can see the full protein, not only in control samples, so that you see only one band, that is the wild type protein. So this is a very big protein. And then in HD patients, we have these two bands because it's the wild type and the mutant one. So I see that the large vesicles, even normalized to the total levels of protein, that these vesicles have more Huntington than the others. Maybe we don't even need to separate them. Maybe if we just have isolation of both together, it's enough as a possible biomarker. But I think that has been my biggest challenge, is to tell people, just think about the methodology that you are isolating the vesicles, how restrictive it is, because maybe you are losing some information. And this has been my biggest challenge. And I want to thank you all for inviting me. It's a pleasure. And I hope that I can give you some of my experience and that it helps your career or future work. Thank you so much.
Thank you, Inês. Obrigada, Portugal. Now, our
Nada.
next speaker is Dr. Martina Fabiano from the Department of Human Structure and Repair, Laboratory of Experimental Cancer Research at Ghent University. And today she will share her journey across continents and research fields and tell us about her experience in the world of EVs.
Thank you very much. I will share now my presentation. Do you see it? Yes, perfect. Okay, well, thank you everyone for being here and Snap for inviting me to this talk. Thank you for the introduction. I'm Martina Fabiano. I am a MD PhD, and I will talk about a little bit about this as well in my career path. From my point of view, it's not so common, so I will recapitulate in this a little bit. So, where I come from. So I am from Buenos Aires, Argentina. As you may know, it's in Latin America. And I studied medicine in the School of Medicine of University of Buenos Aires. And during my studying there in med school, I take immunology course as it is obliged, and I deeply fell in love with immunology. I really liked how immune cells were all over the body, all over the fluids. They were controlling health states and disease states, and I really got into very interested into the molecular mechanisms of everything. However, for medical doctors or medical students, well, research lab work was not that common. So I taught immunology to medical students as well, but that was it. However, when in 2020, when the COVID-19 pandemic started, as part of finalizing my medical degree, I needed to collaborate with society with COVID diagnostics. So I entered the Inverse Institute, that is one of the most equipped labs in Buenos Aires, where I did a lot of lab research actually, and this reinforced a lot the molecular mechanisms that I was very interested on. So I met a lot of people there and a lot of people that were not medical doctors. So I faced a lot of research and I got very interested in that. So I started thinking about doing research. So during this time, I did an internship in Dr. Ostrowski's lab. And soon after I started my internship, I started my PhD. And I did my PhD through 2021 to this till this year in June. My main focus was in plasma extracellular vesicles and immune cells, especially neutrophils and macrophages. And what I did study was how healthy donors' plasma extracellular vesicles were able to modulate inflammation of these two cells, but by two different mechanisms, Gas6 and protein S, mostly for macrophages, and CD39 and CD73 mostly for neutrophils. And how these molecules were located in the plasma vesicles, plasma extracellular vesicles. For example, Gas6 and protein S mostly in the corona, but CD39 and CD73 mostly integrated in the membrane of the vesicles. This work was amazing. I am still really, really glad that I could do it. And I was able to present this work in ISO 2025 this year in Vienna, where I was awarded with the best talk presentation. So it was really a good way of finishing my PhD alongside the publication of my work. So a little bit of where I come from. It's this. And then during my PhD, I was able to do a 1-year research stay in Baltimore, USA, under the supervision of Kenneth Whitwer in Johns Hopkins, where I did a lot of my PhD research there, mostly focusing in the single EV analysis, such as nano-FCM, SPIRIS, or super resolution microscopy, and where I describe better the location of these molecules in the plasma vesicles. So first of all, this experience was amazing scientifically, because I learned a lot from an single EV analysis point of view. But personally, it was a great experience because I was living abroad alone in a new group, in a new lab, in a different city, and it was really great. And having a great group of people was really key for making it a great experience. And this was my first glimpse of why don't I do a postdoc abroad, no? It was a very good experience and I really enjoyed it. So where am I now? Following the map, I finished this year my PhD on June, and soon after I moved to Belgium, Ghent, where I am working with Professor Dr. Hendricks in Ghent University in the Laboratory of Experimental Cancer Research, focusing mainly on bacterial extracellular vesicles. So for me, this was a really huge change because mainly I was focusing in immune cells and now in plasma extracellular vesicles and now bacterial extracellular vesicles and moving abroad. No, so this is where am I now. However, you may ask yourselves, how could this happen? No, so while I will rewind a little bit on my PhD, during 2023, I was able to present part of my work on neutrophils in ISEV annual meeting in Seattle, where I was introduced with my PI by my former PI to Professor Hendricks, and there I talked to her a little bit about science, and I met one of her postdocs. So that was a little bit of that. But after, when I was already during my research stay in Baltimore, I attended to another conference and I met Professor Hendricks again. At that time, I reintroduced myself and we started talking about science again. And at that time, she had like a available position, a postdoc position available, and we started talking about the possibility that I joined her group. So after some talking, well, here I am. It was very successful, and it has been a long journey. Not in years, but it was like a lot, but it was really good, really, really good. So I started thinking to share with you why I wanted to do a postdoc, being a medical doctor. Why did I started a PhD? It's the molecular mechanisms that help me understand the diseases in more depth. But a postdoc abroad, um, well, I always wanted to live abroad, so I thought about the postdoc might be a great opportunity, as I would have finished my PhD. I could change topics, I could change place, so it seemed like a good moment to do so. Why, especially Europe or United States? In my case, Europe. If you know something about Argentina, there's um, a little bit of like limitation on having a state of the art and cutting edge technologies that here in Europe it's less. So I wanted to expand my knowledge and broaden everything and like boost my career. So having access to this technology, it's key for that. And obviously here in Europe, it's easier to have international collaborations, and it's more accessible for growing in the scientific research field. So that it's mainly why, but also there are personal motives. Mainly, I really like the idea of integrated into new cultures and learning from them. And after living in USA and having my research stay experience, I definitely thought so. So I wanted to have that, and also when the possibility of working in the team of Professor Hendricks in Belgium came, it was really special to me because I have family here in Belgium, so it would be a really great change, but I wouldn't be alone and I would be near my family. So it's a little bit of scientific, but also personal stuff. And I can tell you so far, it's been a couple of months, so remember that. But so far, it's a really, really great experience. I, I obviously there are some challenges that everyone face when they change their life, but so far it's been super good. One of the most like important challenges that I've been through, it's like knowing and adapting to everything that it's new here. For example, for me, it's what I talked about before. It's a new lab, a new way of working, a new city, new administrative and bureaucracy steps. Maybe here in Belgium, especially, or again, especially, it's a little bit more difficult to get an apartment, but I got it. So eventually, challenges are fading. For people that like sunshine, maybe Belgium is not that good, but so far it's been fine. And obviously, some scientifically, one of the main challenges is itself the change or the transition between a PhD position to a postdoc position, where you need to not only work with your own project, but also start mentoring and work with the other PhD students. And that is obviously a learning curve that I am experiencing, but obviously there are a lot of advantages, and the main advantage is that people are willing to help you, and you can always learn from other people and talk to other people. For example, here in Professor Hendricks' group, there is a really great group of people that, as I told you, they're super willing to help, and they knew and how difficult it is to get integrated into this new like culture. There are many international people as well, so there is a lot of people who I can talk to work through everything. And obviously, having a research stay abroad helped me to understand what I was going to go through again. And here in this group also, I have like experienced postdocs that I told you before that I already met in the conferences with who are also willing to guide me in this transition into a postdoc. So so far it's been super good. Well, I'm very glad that I can share with you my career path. I'm happy to take some questions, of course.
Hi, everyone. So next I have the pleasure of introducing Dr. Ritu Raj. He did his PhD in life sciences at NIT in India, and he's a fellow Erasmus Mundus scholar, like me. It's great. Where he came to Europe, and he also has many industrial experiences, and currently is doing a postdoc in the School of Biosciences in Aston University, Birmingham in the UK, where he's working under Professor Andrew Devitt. I won't talk much about him now. I think he will take over and present.
Yeah. Thanks, Jonathan. Let me share my screen. Hi everyone. This is Rituraj. I'm thankful for this opportunity to share my experience through PhD, postdoc, and industrial. I started my academic journey from master's to in National Center of Nanoscience and Nanotechnology in the southern area, coastal area of India from Chennai. After that, I moved to central region or towards northern region, did my PhD in life science from National Institute and Technology, Rourkela. During my PhD, I was fortunate to being part of Erasmus Mundus program, and that was my first trip abroad to a very beautiful country, Lisbon, where I had an opportunity to work with Professor Jose and had a very wonderful experience for European culture. I moved a bit around, went to Spain and Paris during that time. And just after completing my PhD, there was an option to do like postdoc industrial job, but I found the industrial job quite easy for a translation center in Bangalore, electronic Silicon Valley of India. And then again did another industrial job where I was introduced with EV liposomes, and it was in Panorama Technology. It's a tissue engineering-based platform in a startup in India. And there I really fell in love with nanovesicles. Starting with nanoparticles and doing nanoscience, it was quite easy for me to transition to nanovesicles and do a lot of characterization and found the translational use of these vesicles. And during this time, I wanted to do more. And again, I started looking for the work where I can be part of more about EVs. And then during that search, I found Professor Andrew David groups, which were working on EVs. It was based in Birmingham. It was a big challenge to move from a industry to back in academia. But when I was introduced, like talked to Professor David, rather than the change, it was the personal connection I had with him. And then with all these challenges, I accepted that, okay, I can move from India to Birmingham and restart my academic journey. A bit of what I was doing before my postdoc, it was not about EV. It was basically about nanoparticles, bacteria. During my PhD, I used nanoparticles and bacteria to make a targeted delivery system for cancer therapy. I did most of the work about the characterization and other stuff. And during my program in Erasmus Mundus in Lisbon, I was again in a chemistry lab where I was working with nanoparticle, making fluorescent particles. The idea was to make a targeted delivery system which can be traced, which can deliver drugs into the cancer. And here I learned most of the stuffs which can be translated in the industry. And then during my industrial exposure at Panorama Technology, where I was introduced with exosomes. And the idea was really fascinating, where we can take the stem cells and collect EV from there, mix with the hydrogel, and create a bioink, which can be used for a lot of tissue engineering stuff, like repairing the diseased tissue, damaged tissue. And we did a lot of translational work, and we are able to showcase to US FDA and other clinical partners. And that attracted a lot of investment also. And during that time, I was also working with liposomes, where we had a challenge to increase the potential of the EV to upload something in that one. So I started working on the liposomes. So in this part, I was able to explore a bit about exosomes and liposomes. And finally, with this experience, I moved to India, to UK, in Birmingham, Aston University, where we did more of the EV stuff. And we started engineering the cells, collecting the EVs from there, and doing tissue repairs, controlling the inflammation. And during this time, I had an opportunity to present our work in ISEF. I was well connected during UK EV groups. So it created an atmosphere to know more about the research, to more about the people, and what exactly going on in this direction in Europe, in US, UK. And that was a lot of stuff during this transition from industry to academia, and learning and creating our own network. And I had a bit of unusual journey. After my PhD, I first went to industry job, and then is being there for quite a 3 or 4, 5 years. Then I came back to the academia and restarted my journey again. So I had experience of both industrial and the postdoctoral experience, and I can a bit of comparison I can do why and where you can do after PhD. Like both the experience has taught me a lot, and there are challenges, and but we can work through that. The mostly during the postdoctoral experience, I found it's great to have international scientific community where you can get exposure during conferences like ISEF, UK EV. I was part of that conference, and it give you a good if you have a very nice mentor like I had a very nice mentor like Andrew David and the entire bioscience group. So it give you a responsibility. You can learn more from your peer, and you can share a lot about them. And another benefit that if you're in a good universities, you have a lots of new instruments, and this field is growing too much, so you cannot have all the instrument at one place. But if the university allow, you can do a lot of work. And another benefit is that it will help you to build your own network. Like you can have your own network through the conferences, through the other scholars. Other benefit, like it help you in grant writing, understanding about the funding. And another thing is that it's the time after the PhD where you can think about where you wanted to go do next. Either you want to stay in academia or you wanted to go to the industry. Similarly, during my industrial experience, it's great to know that the science can be translated. What we do exactly in the lab, can it go to the people? Can this material that we have created, the EV that we have created, can it really solve the problem that we are facing right now? It will make your own a specialist of the field, and it will really like want you to understand how you can translate something from lab to the industrial scale. You will have a boss, and you will be the boss of the others. So it will let you to bring your own team, do your own experiments, take the responsibility. And with all this, it comes a bit of different way of writing about the patents. You will have more information about regulatory investment. So again, it's a completely different setup, and it will give you more financial stability and a time to think that you wanted to stay in the industrial role or you wanted to go back to the academic role. More or less, both have a bit of shift in your entire career. And during the postdoc experience, or before that in Erasmus Mundus, it was a very great shift for me from India to Lisbon, the completely different European, completely different culture from India. But I really enjoyed my time there. And again, in industrial experience, it's a work culture is completely different. What we have in academia, there are tight timelines, deadlines. You have to deliver on time. A lot depends upon the teamwork. You cannot work all alone. And finally, like the work-life balance or the family that you have to support. So the financial stability, both you want in that one. Postdoctoral experiences and challenges usually come if we are moving to a different country. The basically, like for me, being India to moving to UK or Lisbon, the challenges I mostly had about the visa, immigration, the time-bound contract. So that is like always there. So you have to figure out well before that what could be when your contract expires. And back in industry, your entire career growth depends upon how industry behaves. So with my experience, I would really understand that the skill that we generate during our academic learning, it should be transferred, and we should really like look beyond our scientific roles and see that what's going on, and we can build our own network, which is really helpful during the postdoctoral studies and industrial experience, or both. So that's a bit of me. If you have any question, I'm open to that.
Awesome. Thank you, guys. All very different, varied experiences to kind of get a feel from. If people have questions, which we hopefully do, we can put them either in the chat, or if you want to probably raise your hand, you can unmute yourself and then ask a question. And it can be to the whole panel or to one speaker specifically.
Yeah, I can start. I think thank you all for your presentations. I think we need to talk a little bit about it, but to Inês and Martina, after completing your PhD, how did you evaluate whether to stay in the same field, go to stay in academia, or go to the industry? Shift topics or change environments? What do you think in that time?
I think that's something that it's different. I just realized now, because I often forget, is most of the people that I meet in the extracellular vesicles field, all their labs are focused on extracellular vesicles. And my interest is neurodegenerative diseases, so all my labs are focused on neurodegenerative diseases. So the one from my PhD was Parkinson's, and then I did some work in extracellular vesicles. And my two postdoc labs, they are all focused on Huntington's disease biomarkers, therapeutics. So we do a lot of testing of therapeutic molecules, and I brought the extracellular vesicles part. So my labs, they are not focused on the extracellular vesicles, and it can be challenging because I'm the only one working on this field. I don't know, I thought that after my PhD, I would not work in vesicles anymore, but in HD, people haven't been looking at these as biomarkers, that it's a big contrast with what people have been doing in Alzheimer's and Parkinson's, that you have so many publications. So I keep following the neurodegeneratives and disease path, but yeah, extracellular vesicles have been stacking. So I think that there is a lot of potential, a lot of limitations too, depending on how people use the data that they get. But yeah, I think that I never went to the annual meeting, the ISEV annual meeting. I always go to the neuronal one that this year was in Montreal. So I go to the small one, and it was where I met Jolene. I think that this is something that it's a bit different from Ritu and Martina.
For me, changing topic was part of keeping like the academia path. So if I wanted, it was something that I needed to think about if I wanted to stay in academia or move to industry or return to like medicine, like clinical medicine. And I because I really like to be able to help patients or to build up on the knowledge from the other side. I decided to stay in academia, but for really like complementing what I knew about immunology and plasma extracellular vesicles, I thought that I needed a change of topic as well, in order to build up a bigger and broader like knowledge and experience for myself. And this it's something personal that I wanted to, but also if you take into account to like build your own lab eventually, the more that you know, for me the better. Obviously, after all, you need to focus on one only topic, but it's very enriching, and I think that's part of critical thinking and building up on everyone's work. So that's a little bit about me.
Awesome. Britta, did you have anything you wanted to add to that? No pressure. Not.
No, it's great. Thanks. Yeah, I had quite a different trajectory after the PhD. So it's always about like how I can transfer my skill set, because I didn't start with EV, but I was more about nanoscience and nanotechnology. But when I started learning about EV and how it can do all the things, so I come to know that the technique that I have learned during my research, I can use the same. And that helped me a lot during my industrial experience, where I was able to bring new instrument. I was able to guide through, because people are very new to this. Mostly they know cell and gene therapies, but exosomes are completely new. So you need to educate a lot. And the challenges we had when we have the investors meetings and regulatory meetings. So rather than explaining the topic and the EV itself, before going to the application part, that these vesicles can do wonder, they can regenerate the tissues and heal the damaged one. We have to first explain what is EV. They are they are the new thing in the market, but they have been studied for a long. So there are a lot of hesitation there. And then when I came back to academia, so there I was like quite relieved, because it's a niche group where the people know exactly what you're talking about. So there was not a pressure about you need to explain EV, but the pressure about we know quite little about it. People are doing mass spec of EV. They are doing lots of other things that we started up right now. So it's more a learning curve for me. And then I realized that okay, the skill sets are there. We just need to know how we can apply it, and the good understanding and the networking. I really focus on that. Networking is really important, because it's a very small group. Like even we talk about ICEF and UKEV, but compared to the other cell research, EV groups are still very small. There are so much information there. So that's helped a lot. That's it. There are a few questions from the chat.
Yeah, there's a couple of questions in the chat. Yedem, I don't know if you want to unmute and ask your questions. Otherwise, I can just ask them. Yeah, I'll go ahead. So the first one was, how do you manage EV isolation characterization?
You want me to go? Yeah. Okay. I can tell you based on my experience. It's a challenge. It's not easy job rather than about the cells. And we did a lot of ultracentrifugation. We use we use TFF. So we tried all the methods that's like people talk about. Literatures are available. And the key challenges that we had with ultracentrifuge, it's a lot of media. You have to spin them and then collect the very small amount of pellet, and there are chances you're going to lose it. So the challenge comes with that. If there is a high amount of media, you need to concentrate it. We were doing mostly from the conditioned media, not from the plasma. So our challenge was the large volume. And then when we are moving from lab to industrial scale, then we come to know that ultracentrifuges are not GMP compliant. So we have to come back to TFF. That's another challenge. And then we use SEG. So that's how we isolate EV. And the characterization, we did NTA, nano-FCM, flow cytometry. Now the new thing in the market like ONI or nanoimagers, where you can mount them. So there are lots of tools are available, and standard tools like Western blotting, which are still standard. People are using it to do for the marker. And yeah, that's what other advice who are about to finish PhD. I think someone else can take this challenge. The next question.
Yeah. Yeah, if Inês and Martina, if you want to answer the EV isolation characterization, you can. Otherwise, the next question's advice for those who want to finish PhD.
Well, for me, the only advice that I would have is that EV field is very collaborative. So we have a lot of people working on the same fluids or the same condition media or similar condition media. For example, myself work, it's really good to, if you are going to start working with EVs, like catch up with that with myself, because it's really what you're going to do or what you're aiming to analyze on your extracellular vesicles, and which is the fluid from you're analyzing it, the most important thing to isolate them and characterize them. So that would be my answer.
Yeah, I think that it's difficult to have all the instruments at your university. I mean, the probability that you will have, all the instruments to do all the characterization that a lot of people require is really difficult. I would say use the best technique and the best characterization for what you are planning to do. If you want to just work with a special population of extracellular vesicles, then your characterization needs to be very, very detailed, right? But I think that nowadays, you can always argue that you use a certain isolation and you did this characterization, and it depends of the outcomes. Of course, that if you want to use mass spec data or do transcriptomics, your previous characterization needs to be very specific, so you are sure that you are looking at vesicles and certain type of vesicles. But I would say you need to work with what you have, right? And, if you don't have a nano FCM, then just try to do Western blot or try to do some other technique where you can see certain markers and help with the characterization. I think that we can always argue, especially with the reviewers, that, you don't have all the techniques, and we can always collaborate. And like Martina was saying, if you don't have everything at your university, collaborating, it's a good way to just do some extra characterization that you can do in your lab or at your university.
Awesome. And I know Jonathan has his hand up, but there's one more question in the chat that I'll go to first. So Magali says, thank you all. What strategies do you recommend for finding and taking advantage of potential PhD opportunities in EVs for master's students in Europe? The second part is there a specific place where opportunities related to EV research are posted? For example, summer internships or master's thesis projects. For the second part, I think in our SNUB newsletter, we have some postings, but they're typically whoever comes to us and asks. So it's usually PhD or master's or postdoc. But perhaps there's an opportunity for us to reach out to PIs to try to include more summer internships in that as well. But I'll let the rest of you discuss finding and taking advantage of potential PhD opportunities.
I would say going to conferences, it's a great way to find opportunities. And even if the PIs don't have positions open, I always tell students to just start with the email, start the conversation, say that you are interested. Sometimes you can apply for funding without even being there. So you can apply to funding in advance. And then if you get the grant, then there is a chance that you can start at that lab. But I think that conferences are a great place to start, where you can talk with the PIs in person. You can have, an idea of how the PI is, some lab members, or if unfortunately they don't go to the conference that you are going, just send an email, ask to do a Zoom call. Nowadays we all use Zoom, but I would say reach out. Sometimes PI don't have positions open because maybe they don't have the funding, but you can always apply for it. You don't need to be there. So reach out.
I would add that reaching out, as Inês says, it's fundamental, but also doing the research before, no? If you need to know what you are interested in, which PIs work in that, and know what the PIs are working on. So don't appear out of the blue with asking for a summer internship or whatever without knowing where are you going, actually.
Okay, Jonathan, do you want to ask your question?
Does Ritu want to add something to this? Because about PhD positions, I know you did the Erasmus Mundus. Maybe you should.
There are many opportunities during your PhD and before that, even during the masters. Like Erasmus Mundus, one of that. And apart from that, there are other opportunities like DART project and postdoctoral scholarships. Things are there. Like in different country, they have different programs. So that can be always there are website like Find a PhD, Find a Postdoc, which can be, you can browse and find out. And nowadays I see in the LinkedIn, most of the PI started posting their positions and all. But it's really good, like the way Inês and Martina says, that do a bit of research and really want to do, if you're contacting someone, just don't send a mail and say that I want this position. First, look into that if you are really interested in that, or you just wanted to the brand name of that university or the professor. A bit of interest and bit of a background will really helpful. And if you're a master's student, you really don't need to know everything. You're just starting your career, so you are still learning. So a bit of humbleness would be required. So don't say that I know everything. So yeah, that's my advice.
Hi guys, so thanks for the talk. I think the discussion kind of sparked this question in my mind. I think especially what Inês said, and also Ritu was talking about. I do see this kind of disconnect. I'm also a postdoc here. I see this disconnect between EV researchers, fundamental EV researchers, and that being translated into something that's useful in medicine, in any, like even cosmetics and so on, right? So I wanted to maybe kind of spar this kind of discussion I know some of you want to be PI, start your own lab. And I don't know about Ritu, it seems like you want to go back to industry after postdoc, I don't know. But where do you see especially future labs? Because now I feel like EV, for example, biomarkers field in plasma, especially in neurodegeneration, is really popping off. You see labs really focusing. Would you see in the future a lab not just for EV isolation, but a lab that's specializes both in neurodegeneration and EV biomarkers for specific disease? And how does that translate to industry? How do we treat patients better, etc.?
Thanks, Jonathan. It's really right time to ask this kind of question because EV field it's growing very fast. And every day we have new instrument, new information, and it's very expensive. What I can say about rather than the protein antibodies or biosimilars, EV research are not cheap. You need lots of instrument. You need if you are working with patient samples, blood samples, they are really difficult to even When you have the resources, you cannot have the samples to do the research. So it's not easy. And when you have new techniques coming every day, and then you wanted to move towards the translation part, that's become more challenging. Like I mentioned, we during the industrial setup, we work like 1.5 year doing everything in ultracentrifuge, and then we come to the realization we can't do the scale up in that one. So that's like a bit of awareness. We need to know that how it will look like in another industry. It may work in a lab, but you can do a one kit experiment, and that could be very expensive, and you can publish a paper out of it and even do a patent. But when it really need to be translated, it's not feasible. The question comes with the feasibility. And then a second question comes about the regulators, like in US, you have US FDA. In UK and Europe, you have different regulatory. In India, we have different. And these all regulators, they don't want anything fancy. They want simple and concrete. Like the simplest question they ask, are they safe? During the like the ISF and other things, the basic questions were that, like, if you're working with the cancer cell EV, do you think that these EV can propagate cancer to the different tissues? Are they safe to use? If you may find out, like, you know that these EVs are from a particular cancer cells and you have established the pathway, but how you're going to use that, how that information will be useful for the patient. So those questions are still open. And when we were working with the stem cells derived EV, we had a like the precedent that the stem cells are good to use, but our hypothesis was that we are providing something cell-free, which are not going to divide. It's like another inert particles. They will stay as it is and just give the functional benefits. And but the characterization tool, like the thanks to ISEF, when they started publishing the basic criteria to how to characterize EV, how to do the nomenclature, that was really helpful from I think 5 or 6 years back. There are so many ambiguity about the nomenclatures, about the tools that we are using, about the markers that we are using. And so it's give a kind of a like a protocol setups where you at least do the minimum. Like if I'm doing the EV characterization, the minimums are to tell the size, what size they are the EVs or not, to do the basic minima about the tetraspan and all. So now these things are a standard practice now. The second things comes the biggest challenge which come to us, how you're going to give it to the patient. It's maybe a large volume of media, but patient doesn't take the media, and they need something like that. You take a paracetamol tablets, it should be fixed. It should always show you the same effect again and again. So the questions were asked, are they reproducible? Can you make the another batch with the same effect? So these were the difficult question to handle, but we started putting more and more characterization to prove that our EVs are safe. They can produce the same effect. So, and then we come to the realization, everything keep on adding up. Like, if I'm doing a NTA, do I need to do a nano FCM? Do I need to do some other tech characterization to prove that these are the same EV that I'm talking about? And the same way, so then the conversation happened, and then we come to know that it's easier to do the basic minima, and then go to the market and tell the people. And like the groups like us, we had another responsibility to teach to other scientific community. I'm not talking about the non-scientific community. I'm just talking about the scientific community about EV. There are still people, they don't believe that they really work. They are saying down, it's something coming out of the cells. It's not what you're saying. It's always there. So we had a good understanding. We are not just saying that we are telling that so it should be work, but we have to prove that and tell that no, it's something beneficial and it's a really good value to it. Yeah, I hope I answered, or maybe I just carried away.
No, I asked a very loaded question. I'm sorry.
Yeah, no, it's a long feel like understanding. I have seen it, and so I can relate to that question, and I'm still working on that. And your second question about moving back to academia or industry, for pretty, I'm still unsure. Like academia give you a sense of belief about science where you can do more and more, but it's kind of non-ending. But industry put a pause on you that no, this is the it you need to deliver. Bring something which can go to the market. So that's the major difference I can see between academia and industry. Yeah.
Awesome, thank you. I know we're 2 minutes over the hour, so if anyone needs to leave,
Sorry.
for sure leave. Inês and Martina, I do want to give you an opportunity if you wanted to speak to that question.
I agree with Rito. It's very difficult, and I think that a lot of people that publish in vesicles, they don't think about translation, how your work that you do in the bench, in the lab, translates to clinic. And I know that a lot of companies are interested in vesicles, but the fact that people can't reproduce other people's publications, it makes industry very frightened. And I think that it's something that they are not really willing to go because they can't reproduce whatever other person published, right? And then you have these huge data sets that you don't know what to do with them, right? They say, oh yeah, I saw, I don't know, 20,000 microRNAs and these 10 change. Yeah, but then how are you going to measure that in plasma or in CSF? And I think that the only way to do that is if your lab actually already has collaborations with industry, you can get their input and you can, maybe try to work with them. If your lab doesn't have collaborations or if it does a lot of molecular biology, then it's really hard, right? I mean, the only way to make your work pass that barrier from the bench to the clinic, it's really if you collaborate with industry, and that we can't do anything about it, So I think that side, it involves a lot of discussions and looking for industry partners that are willing to work with you on that step. But I think that's a big problem, the fact that we can't reproduce a lot of things. And how are we going to establish that? How are we going to use vesicles, or how do we know which hits to look for into the vesicles? And that is another question. And yeah, the question between academia and industry. So I have been changing. Initially, I want the industry, and now I want academia. But I still like industry. I think that you need to have, if you like biomarkers or therapeutics or this part, you need to have experience from both sides. But I want one thing, but it doesn't mean that it will happen, right? So I'm open to other opportunities. So I will go and see how it goes. Unfortunately, science nowadays, it's really hard, especially getting PI positions, because a lot of people want to. And me personally, I'm not restricted to a specific country or continent, and I think that helps me. But of course, that I understand that people want to be in their own country or they want to be closer to their family. And of course, that if you restrict your positions to a certain country, it will be really hard. But it's not impossible. I think that it also depends on when the positions open, and keep trying, and connections. I think that nowadays having good connections is just the best help that we can get.
Thank you. Martina, do you have anything?
Okay. But very short. I think like coming back to the translational research, I think we also focus a lot on like industry, but at one point we need to go to the other side of translational, and we need to collaborate a lot with the medical doctors and to see what patients that suffer the diseases that we study really need. For example, getting biomarkers, it's crucial. And for example, for detecting the disease or detecting the progression of diseases. But if we don't know what it is lacking in the field, we won't be able to do more for that. Industry or academia, well, I don't have the answer, but I think there's pros and cons at every side. So it's a balance of what you want to do and everything.
Awesome. Thank you guys so much. And yeah, I think we really need to, we've talked a lot about collaborations with industry, but we also need to consider, collaborations with the clinical side with both doctors. Are they going to administer it, and are the patients willing as well, right? So thank you three for a wonderful discussion and for all your time and staying a little bit late. Jonathan and I think Barbara had to run to a meeting. Jonathan, do you have anything you want to add before we take off?
Thank you, guys. It's a pleasure.
Thank you so much.
Thank you. Thank
Well,
you.
thank you very much.
Everyone.
Bye.
Bye.